Thank you, CC. It's a real pleasure to be here, and I want to thank the organizers for inviting me. Now, these are my learning objectives. What I'd like to talk to you about is a story about how we got here. About 30 years ago, almost to the day in December of 1984, a group of elite geneticists from the United States, among them George Church, Lee Hood, Ray Geslin, and others, were at Alta, Utah, and conceived of the idea of sequencing a human genome in 1984. Nearly 20 years later, they achieved this moonshot, as illustrated in this press release from Francis Collins and colleagues. And Dr. Collins called on us to join as a scientific community to realize the full benefits of having a genome sequence. So that's probably why we're here today at a conference like this, to really think about how we can take sequence information and other aspects of genome technologies and really apply them to healthcare. About 3 months ago, the Personalized Medicine Coalition put together a report of personalized medicine by the numbers. By their reckoning, we've achieved in the last 6 or 7 years a 10-fold increase in the applications of genomics and genomic medicine in healthcare. You can see some of the other statistics here. I'm not going to go through them in detail, but one of the interesting ones that I'm sure you're going to hear more about tomorrow is the fact that FDA drug labels now contain genomic information in around 155 products. If you contrast that to the time that the genome was sequenced in 2001, there were only 4 labels that contained any information about genomic-guided therapy. I like to use this diagram to illustrate where the applications are from womb to tomb of genomics in healthcare. This red line illustrates the course of chronic disease from birth ultimately into death. And the treatment paradigm today is to treat people at the right-hand side of this curve when the burden of disease is high, the costs of care are high, and the chances of reversibility are quite low. So obviously we want to shift the paradigm of care to the left. And we can do this through a number of technologies today that just weren't available even a decade ago. We can now do prenatal sequencing at the time of conception nearly. We can do newborn screening at the time of birth using sequencing and other technologies. A suite of molecular technologies are now available for screening for diseases in high-risk individuals, and new molecular diagnostics and prognostic tools are being generated in laboratories such as in this institution and others every day. And of course, pharmacogenetics is now giving us a toolbox to use for making decisions about appropriate therapy to optimize the benefits and reduce the risks. We've seen evidence, particularly in the last couple of years, of the proliferation of targeted therapies. And it's not just cancer. The first example on this list, ivacaftor, has been approved for patients harboring a particular mutation, the G551D mutation in the CFTR gene, for patients with cystic fibrosis. And for the first time, those individuals have a chance of better quality of life and longevity than ever known before. And since the approval of this drug in 2012, another 8 mutations have been shown to be effective and now are included in that drug label. You see a number of other examples that are quite important over the last several years. Crizotinib was approved in non-small cell lung cancer for patients harboring the EM4 mutation, showing a dramatic response rate in clinical trials. And recently, a follow-on drug, ceritinib, has been approved for the same indication. Vemurafenib and trametinib for various types of melanoma harboring specific mutations give those patients at least a chance for better quality of life for a period of time. And recently, there was even a novel indication for a new molecule for metastatic breast cancer targeting the HER2/neu receptor. So we see a proliferation of these surgical therapies, these targeted therapies coming to the marketplace. And it's not just genetic variants that are at the root of our genomic medicine paradigm. There have been a number of tests developed using the express genome. Several of them are listed here. For several years now, probably since about 2006 or 2007, women have been making treat or no treat decisions for breast cancer using a gene expression test, the Oncotype DX test. And in cardiovascular disease, as you can see on the bottom of this slide, There are tests such as the AlloMap blood gene expression test that is used routinely in transplant centers to obviate the need for invasive endomyocardial biopsies, or the Chorus CAD test, also a blood gene expression test that measures the degree of coronary disease burden and that in some cases may obviate the need for invasive and costly coronary angiography. So as you can also see from this list, several of these tests have been FDA approved, and several of them are also reimbursed by third-party payers. And we can also note that there are a number of gene expression tests in the pipeline for tumors of unknown origin, for Alzheimer's disease, for squamous cell carcinoma, for lupus, breast cancer, idiopathic pulmonary fibrosis, and work that we've done in our laboratories using blood gene expression testing to diagnose viral versus bacterial disease based on the host response. So I think we can see a future in which more and more of these express genome technologies will be playing a role in clinical medicine. Now, the first, I think we can benchmark the role of genetics in the understanding the etiology of chronic disease back to 2005, not that long ago, when the first genome-wide association study was reported in the literature. This was for age-related macular degeneration, and groups from Duke, as you can see in the upper right-hand corner of this slide, as well as from the National Eye Institute at the NIH, reported that a variant in a protein gene called complement factor H was responsible for AMD. The first time this pathway was ever known to be associated with this disease. And also, it's interesting to note that today there are therapies in the pipeline that are targeting the CFH pathway for AMD. We fast forward to today, we now see literally thousands of genome-wide association findings have been found across hundreds of chronic diseases and complex traits. So this is really a stunning accomplishment, probably one of the more stunning accomplishments in biology, to look at the underpinnings of disease in an unbiased way and giving us insights into mechanisms that have never been hypothesized to be at the root cause of these diseases before. But as this slide says, it's also disappointing in that the odds ratio of carrying the risk allele for many of these diseases, or most of them, has been less than 2. Making these not necessarily sharp tools to use in the clinic for predicting disease risk. Maybe we've leaped over a very important tool that has been available to us for decades, literally, probably our lifetimes, that is family history. This is a very nice study from the UK, from Nadeem Qureshi's group, showing that in a pragmatic, cluster randomized controlled trial, that implementing a family history intervention in a primary care clinic can identify individuals, give you a 40% increase in the identification of individuals at higher risk for cardiovascular disease over a control cohort. So why aren't we using family history more often to ascertain individualized risk? At Duke, we've created a computerized software platform that allows for patients to enter their own family history. Often the barrier is that the clinicians simply don't have the time to collect an adequate 3-generation family history. The data entered by the patient is subjected to clinical decision support rules, and at the time of the visit, the patient has a report of their risk, the physician has a report of their risk, and it's a very productive interaction between the patient and the physician to determine what the next course of action should be. We can't ignore that genome sequencing is clearly an important component of our armamentarium going forward. The first human genome cost nearly $3 billion to sequence in 13 years, as illustrated in the upper left-hand corner of this slide. And there's been a precipitous drop in both the cost and the throughput of this technology. And there's prospects of a USB-driven sequencing platform that will come out this year that could certainly continue to revolutionize this technology and make it reasonably accessible to almost any of us. This precipitous drop in the cost of the technology and our comfort of using it has led to a number of large-scale genome sequencing initiatives that are illustrated here. Genome England, Prime Minister Cameron has committed the funds to sequence 100,000 UK genomes and integrate those with the National Health Service health records. In Saudi Arabia, a collaboration with Life Technologies focused on cardiovascular disease is also going to sequence about 100,000 genomes. The Geisinger-Regeneron collaboration announced earlier this year is going to integrate genome sequence information with Geisinger's EPIC EMR. And more recently, the Qatari Genome Project has been announced to sequence 300,000 native Qatari genomes, also linked to the Cerner EMR system in that country, all of which I think are aspirational goals to demonstrate that the value of genome sequence linked to electronic health information will improve the quality and reduce the cost of healthcare in these countries and beyond. One needs only to be a student of the New England Journal of Medicine to appreciate the role that sequencing information has had on individuals. These are some infectious disease reports showing that real-time genome sequencing in the upper left-hand corner was used to identify the source of methicillin-resistant Staph aureus in a neonatal intensive care unit in the UK. At the bottom left, the transmission events from camel to human of the MERS coronavirus in the Middle East was also ascertained through sleuthing using genome sequence information. And the young boy with an undiagnosed febrile illness was found to have leptospirosis, and a life-saving antimicrobial therapy was given to him on the basis of sequencing results. We've also seen applications in numerous areas. Just a couple are illustrated here. The benefit of sequencing for early detection of cancer, superior to that of the currently available immunohistochemical rates using fecal DNA sequencing. And in the lower right-hand corner is one of the first case series of undiagnosed rare diseases demonstrating a diagnostic finding in as much as 25% of the cases evaluated. And importantly, out of 120 some odd cases that were presented to insurance companies for sequencing, 90 to 95% of them were actually paid for. This is actually the reason why we have to do genome sequencing. It's cases like Nick Volkers, who I'm sure is familiar to everybody in the room. Medical College, Wisconsin, was able to sequence Nick's genome and find an actionable mutation that ended up saving his life. And it's these lives saved on the basis of understanding the biology and where therapies could come to bear, I think, is what drives us to really make this a much more tractable platform for routine use. What we've done over the last several years is successfully push the bottleneck downstream. We can certainly generate high-quality data. We can process and align the reads. We can create the annotation of the genome. But what it comes down to is genome interpretation. What do the sequence variations mean? Do we have sufficient reference genomes? And importantly, who is going to deliver this information to the patient, and what context will they provide that information in? I think all of us in the room probably would agree that we don't have the workforce sufficiently trained to take on this type of complex information, at least today. And that's one of the major challenges I think we'll face going forward. The Harvard Partners Brigham and Women's Hospital MedSeq program, part of the CSER consortium, which stands for Clinical Sequencing Exploratory Research from the NIH, has actually created a one-page report for genome sequence. Now, I know you probably can't read this, but the first page gives you information about monogenic diseases, about carrier risk, about pharmacogenetic risk, and blood typing. And then the back pages gives you sufficient detail to understand where those summary information came from. So I think it's conceivable that we could make a report for a whole genome that will actually deliver just-in-time information to the clinician so they can have that important conversation with the patient. It's clear that cancer genomes have been ahead of our germline genome ability to both deliver important information. And this really nice review from Bert Vogelstein last year, he and his group summarized the fact that probably, at least as of today, there are about 140 genes that are known to be drivers of the oncogenic phenotype. They're very closely linked to important disease mechanisms that support oncogenesis, cell fate determination, cell survival, genome maintenance. And it's really important to recognize that genetic heterogeneity exists and can impact the response to therapeutics. It's not completely clear, at least to me, how we're going to get away from genetic heterogeneity, but it's a fact that tumors are genetically heterogeneous. And also importantly, the oncologists have not only access to the somatic genome from a patient's tumor, but the reference genome they use is the germline genome, the reference genome that they use to determine which variants might be active in a cancer is the germline genome. So they also have the ability to make decisions around patient care that also emanate from germline genome information. And I think it's going to be an important opportunity for the oncology community to bring these two really important pieces of information together going forward. Oncology also has changed the landscape of our vernacular for even describing tumors. 20 or 30 years ago, we could describe lung cancer on the basis of histologic phenotypes and location, perhaps. Now, 20, 25 years later, we can subset at least the adenocarcinomas of the lung into a suite of molecular phenotypes that are described by the underlying genetic variation that are driving that particular tumor. EGFR, KRAS, EM4, ALK, et cetera, are ways that we can now describe these tumors. And importantly, if a patient happens to have one of these mutations, in many cases, there's an FDA-approved drug, a targeted therapy that could address that patient's underlying biology. And in other cases, there are clinical trials ongoing with experimental therapeutics that also might be able to address that patient's underlying biology. So patients today with a cancer diagnosis have numerous opportunities that were not available to them before, either to go on FDA-approved medications or to be enrolled into clinical trials with a chance of having a therapy that actually might target their particular underlying genetic variant. Now, if you look through academic centers such as Mayo, Duke, Dana-Farber Cancer Institute, it's likely that most patients coming to one of these centers will have an actionable and targetable mutation, and that will be acted on. If you go into the community, and survey data from ASCO has shown that only 5% of patients with lung cancer in the United States actually get EGFR measurement in the first place, the standard of care. So there's clearly a knowledge gap between the places that are promoting and facilitating the discoveries of these and the practicing community of clinicians and oncologists in this case. And we need to somehow fill that knowledge gap and really enhance the diffusion of information from places like Mayo and Duke and Dana-Farber, MD Anderson, and others to community-based practices so all patients will have the benefit of this technology. In a commentary I wrote earlier this year, published in Nature, I advocated for health systems to take on a significant role in this arena. One is to make sure that our clinicians have the up-to-date information, just-in-time information, for adequate decision-making around most decisions around that might use genomic information. And also to be active participants in the clinical data that needs to be generated to build the evidence case that genomics actually provides value to the patients, providers, and the health care system, the so-called learning health system that Dr. Huddis mentioned earlier. And this is just a schematic diagram, at least of the simple elements, not so simple to do, but simple in concept to bring together to make this happen. The electronic medical record, genome sequence information, family history information, and a knowledge base that can be used to make comparative analyses that will drive the appropriate clinical decision supports, illustrated by this box called the IT system, a not so trivial part of the infrastructure to build on. But the NIH and others have taken on this challenge to some degree, and the Electronic Medical Records and Genomics Network, the eMERGE network, is now in its, I think, 12th year of doing this type of work. And you can see the various centers across the United States, including Mayo, that have used the linkage between EMR data and genomic data to do novel GWAS, genome-wide association discoveries of electronically medical record-defined phenotypes. They've developed the consent methodology. They've engaged the community. They've addressed some of the privacy issues. They've also branched into pediatric populations. You're going to hear more about the eMERGE's pharmacogenetics work probably tomorrow. And certainly they're developing strategies for patient and provider education as well as clinical decision support. Illustrating one of those clinical decision support opportunities is on this slide. This comes from the Vanderbilt site, and this is a clinical decision support window that fires when a provider orders a drug called clopidogrel, which is used to prevent clots in patients that have undergone a percutaneous coronary intervention. In this particular case, it's known that the patient harbors a mutation that will prevent them from adequately metabolizing the drug and achieving the full efficacy of this drug. So it tells the physician that, and it also makes suggestions for alternative therapies that the patient might benefit from. And if the physician decides to not use those therapies, it also asks them why not, so that it begins to learn why there are cases in which this might not be the case. So this is just one example of a proliferation of clinical decision support engines and rules that will make the learning health system a reality and also help both the clinicians and patients digest and integrate a lot of complex genetic and genomic information. The other thing that the eMERGE network has done is it's formed the Clinical Pharmacogenomics Implementation Consortium. This consortium has now delivered and published 12 guidelines for commonly used medications and essentially an instruction manual or manual of operations of how we might actually begin to think about thiopurines, tricyclic antidepressants, statins, and other cardiovascular medications, where there's an opportunity to look at the patient's underlying genetic background and make better choices and better decisions about what drugs might be best for them. Now, some of you may have seen the formation of a company called Iodine. It's an interesting concept from Tom Getz, who was the former editor of Wired magazine. He's crowdsourcing drug information in Iodine. So you can go to Iodine and look up any drug and find out what patients are really experiencing with taking these medications, whether they're achieving a benefit, whether they're having adverse events, what those adverse events are. And this could be quite a game changer in terms of our understanding truly of the full effect of drugs, particularly once they hit the marketplace and we have little ways to really capture what's happening at the patient level. I also wanted to highlight the fact, as I did earlier, that the NIH is playing a significant role in trying to advance genomics into medicine. There are these 4 networks are critically important, certainly not the only solution, but critically important to the solution of implementation of genetics and genomics into healthcare, and also ascertaining the outcomes of providing that information. I mentioned the eMERGE network. Last year, we began the IGNITE network, implementing genomics in practice, which is a series of genomic medicine demonstration projects using pharmacogenetics, using genetic risk information to prevent chronic disease, as well as family history. The Clinical Sequencing Exploratory Research Network is also looking at a whole genome and exome sequencing, both for tumors as well as germline, as well as in newborns and adults, to begin to understand how do you implement genome sequencing into a medical environment and deliver adequate decision-making. And the longstanding Pharmacogenetics Research Network, of which Mayo has been a member since its inception, also not only discovering drug-gene interactions, but also their implementation into healthcare environments. So in the last few minutes of my remarks, I wanted to talk about a data convergence that I think we cannot ignore. This is a quote from the chairman of IBM, who estimates that there are now 2 billion of us that are connected by the internet, and that probably a trillion, the Internet of Things, constitutes 1 trillion devices that in some way might be able to talk to each other. And the question becomes, can we harness this data and put it to good use in this concept of precision medicine? I think it's worth mentioning that cell phones connect us all. This is a little dated. There are probably close to 7 billion cell phone accounts across the globe. And in many cases, they're not simply used as communication devices. They're used as ways to deliver important medical information, to remind patients about being compliant with medications that they're on, to remind patients that they should exercise or adopt certain lifestyle changes to mitigate their risk for diabetes or heart disease. So cell phones are, in some ways, an underpinning of, I think, an extension of our healthcare system in ways that allow us to communicate more effectively treatment decisions. And also, it's a two-way conversation. So this speaks to the notion of Moore's law. What you see on the left-hand side is the fact that compute power doubles approximately 18 months. And you can see the curve below Moore's law on the left-hand side is the way that sequencing technology has changed and has far exceeded Moore's law in its capacity. On the right-hand side is Metcalf's law. Metcalf's law says that the power of a social network is the square of the number of users. So how do these two things come together? And this is just one study that illustrates this. It's the notion that we can take social media or self-reported phenotypic information, not through a clinical trial, not through a clinical research study, not even through an EMR, but self-reported information, and link that to genetic information and begin to recapitulate a number of the GWAS findings that were done through those types of rigorous studies in the past. So it really speaks to the power of us and our patients as adequate reporters of information that could be quite important for us to really understand the underpinnings of disease and certainly understanding of health. And the other thing that's happened in parallel is this proliferation of sensors. Now, one might hear the word, when I hear the word sensor, I also hear the word phenotype. For the first time in history, we have the ability to actually do continuous monitoring of very important clinical phenotypes. So you remember the Borg from Star Trek, those of you who are Star Trek fans. The Borg was a cyber community of individuals whose collective intelligence was greater than any one individual. And if you think about the quantitative self movement, some of you may be part of that, where people that are meticulously recording all the information about their health and well-being ad nauseum, this might be the beginning of a Borg-like community for healthcare that delivers big data and data that we can analyze to really understand different patterns of health and disease. And it's manifest in things and products that we see every day, in Fitbits that one might wear around their arm, or around their neck, or around their waist, or their Bluetooth-enabled scales, or the new Apple Health Kit, which is now monitoring those of us who subscribe to it on a continuous basis, a number of aspects of health. And there's just sheer proliferation of products probably driving this to be a billion-dollar or multi-billion-dollar industry over the next few years. We've seen an amazing growth in technologies that allow us to sense very important biological traits. So the Google glucose sensing contact lens is illustrated in the upper left-hand corner of this slide, and on the right-hand side is another contact lens that senses intraocular pressure. So it allows the patient now to begin to understand better their traverses from a managed disease state to potentially an unmanaged disease state and take action. We've also seen the proliferation of very low-cost sensor technology. Paper-based sensors can be produced at fractions of a penny and can be delivered to the developing world. Can you imagine that we had a technology that could measure proteins in the nasal lavage or nasal secretions that would be captured in something as simple as a tissue, and the tissue might turn a color, indicating that you're about to come down with an upper respiratory, viral, or potentially other type of pathogen-based disease. So these smart tissues are not far-fetched and could actually have a very important impact on things like Ebola and other types of transmissible diseases. We should not also forget that the social media and is a very powerful tool for understanding health trends. And this slide illustrates data from several years ago showing how people search the internet for terms related to the flu. So do I have the flu? Do I have a fever? What are cold and cough remedies? And you can see this cyclical spike each year that coincides very nicely with the flu season. So the way we search the internet is actually a way to think about a Bayesian prior probability of actually one might having that disease if you had that data available to you. And the same thing can be done with Twitter data. So several groups have monitored Twitter feeds for similar types of statements by individuals about flu, fever, sore throat. And in the red lines here, the red dots indicate the models for predicting that somebody has the flu versus, or at least the frequency at which somebody reports those symptoms, coinciding with the black line, which is the percent of influenza-like illness that's reported in the local community. And we can also think about gamification. Gaming programs, while entertaining, also might provide quantitative information about cognitive function, about mental performance, and other aspects of mental health. And they're readily quantifiable, and in many cases, they're being continuously monitored by the groups that have invented them. So we see a summation of a number of different data streams, which, if brought together, might give us a better representation, or perhaps a more precise representation of health and disease states. So this notion of an integrated data repository does not exist today. We're only getting close to bringing EMR data and maybe some genomic and imaging data together. But imagine if we could bring geospatial data, social networking data, sensor data, and these other data streams together into a mineable and accessible data repository. Then we might be able to take the analytics that we're developing today and discover new aspects of biology of disease for sure, but also to maybe redefine how we classify disease and, importantly, predict what might happen in the future. So this has implications for how we even design studies. And this is a view of what 21st century epidemiology might look like. Longitudinal cohort studies that look at people over time using a variety of physiologic imaging, as well as genomic and other types of tests that will use a systems biology approach to quantify health and disease states. And we'll also obviously need to engage the patients and provide them with information that's meaningful to them. We need to validate these wearables and the devices that I just talked about and use them in more constructive ways so that we can build the types of models that I alluded to. So what might this look like if we were to be successful? And if I can figure out how to, can you run the, there we go. Just have a short video clip here just to give us a potential view of the future.
Technology has given us an unprecedented window into the human body. But on a day-to-day basis, we're still in the dark about our own health. We are changing that.
You okay? Do you feel okay?
What if, instead of fearing the worst when you notice something out of the ordinary, you could identify the condition yourself? Getting the right diagnosis would save you worry.
It says it's roseola.
And an unnecessary doctor's visit.
Rest at home. It's okay.
Instead of hearing about a viral outbreak on the news, imagine you got an alert that was tailored to your family's needs. It would also give you advice about what to do next. What if you had a way to identify what was wrong right away?
It says it's 103.8.
A way to get all of the information you need to understand the situation. And in serious cases, you would know when and where to seek help.
It recommends taking it to the urgent care.
We're building a way for people to check their bodies as often as they check their email.
Check your bodies as often as you check your email. What a concept. But I think this is quite a visionary video from Scanadu. I don't have anything to do with Scanadu, but I really subscribe to the notion that these types of technologies will converge. Whether they converge in 5 years, 10 years, or 20 years still remains to be seen. But I would argue that to realize the opportunity in genomics in medicine and healthcare, there are these game changers. One is sequencing. It is here to stay, and is transformative, both for clinical applications as well as for how we do research. We need to subscribe, if we're not already, to data sharing and increase that liquidity and fluidity of data across the silos in which it currently resides. That is the way that will facilitate the validation of our scientific findings and ensure the integrity of those results to some degree, and also accelerate the science. The learning health system model that I mentioned, Dr. Hudis mentioned, this is a critical need. A question might be is, what is the motivation for a health system CEO to actually make that type of investment? It's not completely clear, but because there are significant investments that need to be made to build the infrastructure needed to accomplish the vision for what learning health systems need to be. We should embrace e-medicine and m-health, but also be cautious about them, because right now I think the technologies are at a very early stage. And like a lot of other technologies that we use in medicine routinely, these need to be validated both analytically, clinically, and be shown to be useful to bring them to the mainstream of healthcare. And importantly, the patients. I mentioned a few examples where crowdsourcing might play an important role. And I think our patients are an incredible resource. We just need to make sure that they remain in the center of focus, that they are engaged and feel empowered to participate in our research agenda. Thank you.
Dr. Ginsburg, thank you for that. You've gotten a lot of questions already. They've been coming in, so thank you everybody for sending these along. And we're going to try to get to as many of these as we possibly can. I'm going to start with a really simple one, if I can even read this question. Why is there a discrepancy between leading research institutions and other medical institutions when implementing genomic tests? And they give an example here of the EGFR in non-small cell lung cancer. Do you think that it has to do with cost, education, disagreement?
I think it's a combination of factors, and I'm sure that there are people in the audience here today that come from community-based hospitals, and I would certainly want to make sure that they have a chance to weigh in on this, but the community-based practitioners that I've talked to speak to one, lack of access to the technology, that it's often not brought to them in a timely fashion. Certainly, there's a lag between publication of something that's even in a guideline and adoption into practice. Some of us call that the T3 research, translational research barrier. That's the diffusion of innovation, which has been around for a long time and is not unique to genomics. Secondly, some of the concepts that you just mentioned, Ceci, education. And I don't mean that the practitioners in the community are not educated, but it's that keeping up with the current guidelines, keeping up with what's happening is a constant challenge for all of medicine. And again, it's not unique to genomics. What are the types of educational seminar series or grand rounds or other, what is the best way to make sure that this type of information is transmitted effectively, and efficiency has been something that has challenged us for a long time. And then the issue, the last issue might be even cost and reimbursement issues, that they haven't been negotiated locally as well as they might have been in other places, or there's not the research infrastructure, which often is paying the freight on some of these novel molecular tests.
Well, it seems like it's been a little while now, but I think I've certainly read the statistic that it can take 7 years for a new medical insight to reach practicing physicians. I don't know if that's still the case.
That's optimistic.
That's optimistic.
I think the statistics that I have read and heard about is from idea to dissemination is about a 14, 15-year time horizon, believe it or not. That's not just in things that come out of, in inventions that come out of the medical centers like this one, but also from the pharmaceutical industry. You know that it takes 13 years or 14 years to develop a drug and $1 billion to do so. So there's just that lag is highly prevalent.
So let's switch over to the topic of sequencing for a minute. Do you think that I should be sequenced? Should everybody in this room be sequenced? Should we all?
If we harken back to the last talk and think about cost, I can think of no better way to skyrocket medical costs than to recommend that we all get sequenced, not just because the technology itself might cost something, but the things that we would be chasing, the incidental findings, if you will, the things that we don't understand, would add additional tests and additional a cost to the system beyond our capabilities of imagination. So I think we have to take a step back and think about sequencing as a real opportunity for selected patients. We need to define how we select which patients are going to be the most benefited by sequencing. And those groups that are doing rare disease genomic sequencing in particular have done a great job in thinking about what are the attributes of a particular case that would pull the trigger on a sequencing activity, and maybe who may not need it quite yet. So I think it's that ability to, like any other test, to develop a sort of a decision tree that allows you to arrive at a conclusion that sequencing might benefit this patient versus someone else. So yes, sequencing might become more routine, in certain cases, such as focused sequencing for the newborn. And one area I guess I would advocate that we think about is pharmacogenetics. Now that doesn't mean sequencing one's entire genome, but really focus genotyping or potentially sequencing a small set of genes for drugs that it's highly likely that we will be taking in our lifetimes. That will be information that we will carry with us forever, and we might as well use it at the earliest possible time.
And so would that happen, do you think, on an individual patient type level or a population health level? I mean, let's say, for instance, we know that diabetes, high blood pressure, there are some conditions that are incredibly prevalent in our society, and sadly, the trajectory seems to be going up, not down. So are you suggesting at that population health level it might be a smart investment?
I would say that you're absolutely right. We need that if we could identify individuals that are higher risk for certain chronic diseases like diabetes, heart disease, or maybe people that are higher risk for diseases like cancer, these might be the individuals that we might selectively choose for things like pharmacogenetic, genotyping, or potentially sequencing pharmacogenes. And whether that will translate into a population health benefit, I think, is something that we should think about how to study. So I'm not going to go on a limb and recommend that we all now get our gene sequence in that way and pretend that we'll achieve a population health benefit. But I think there's a likelihood that it will translate that way, and we should design the studies to prove it.
So I bet Pat Garrity, who's going to be up a little bit later, is sighing a little relief about that, because he's probably imagining the cost of all of this right now. We'll get a chance to ask him a little later today. But from your perspective, doctor, what could be the role of insurers in some of what you've presented today, as well as, for instance, the pharmaceutical industry? How can they contribute to this?
Yeah, I think one of the things that I've experienced, and I think many of the investigators doing genomic studies in the room probably have experienced, is that we all design our studies with our sort of pointy-headed academic ideas about what should be the end game that's going to be important to measure. And we go to an insurance company and we tell them about our study, and they say, well, that's not something we're really interested in measuring, and it's not going to drive us to make a payment or coverage decision. So what I would encourage us to really be thinking about is to bring the payers into the discussion as early as possible when we're designing some of our studies or some of our paradigms for care even, and engage them in that conversation. They can't be something that's just left to the end, because at the end you'll be sent back to try to redesign it and repitch it at some later date.
And isn't that increasingly also the case or the potential risk for pharmaceutical companies in terms of going very far down a path without having some of those conversations around payment?
I would say certainly that the smart pharmaceutical companies, of which I'm sure they all are to some degree, are not only engaging the payers, but also the regulators in very early discussions, particularly when it comes to these targeted therapies or companion diagnostics. It's not something that just happens organically. There's got to be a highly orchestrated series of processes and relationships with diagnostic firms, with the FDA, and with the payers to make sure it will all come together to a solution that will work for the company as well as work for providers and payers and healthcare systems.
I liked that video clip so much, and at our Health Research Institute, we do a lot of consumer research, and we are hearing in record numbers consumers that are saying they want much more convenient care options and lower costs. Those are two at the top of their list. So for instance, when we ask consumers if they'd like an at-home strep test, 60, 70%, yes, we want it. But there have to be some clinicians sitting here in this room right now who might have gotten a little nervous watching that video, because are we all going to start playing doctor at home?
I guess when I look at that video, I see the front end, but I don't see the back end. And the back end is a tethering of the types of technologies that we see in that video to healthcare systems and provider teams. If you think about Apple, Apple is now integrating their healthcare information into Epic. So it's not something that's happening in a completely isolated silo away from the healthcare system. It's really trying to drive it to be integrated. And I would argue that the success of the technologies that we saw in that video are going to be critically dependent on their integration with healthcare system providers, as well as their workflows and data, that it's not going to be, it's got to be tethered.
And does that also tie in with, you mentioned the ability, sort of the one-page report for the clinician. Is that part of the appeal, the integration of all of that information into one good snapshot?
So providers, I think, want to be able to have the best and most efficient and quality interaction with their patients. They don't want to have to spend their time on a computer looking up something and not having that discussion with the patient. So to the extent that we can provide just-in-time, red light, green light, terse reports for decision making, and also continue to give them the precious time that they have, make that the highest quality. That's what I think most providers will be looking for.